All right, welcome folks. This is part 2 of the Varroa Mite IPM series hosted by the Northeastern IPM in conjunction with Jennifer Lund and Kim Skirm. And you can see Kim has been very active over the weekend, creating a very cool background. And I am Jana Hexter. I work for the Northeastern IPM Center, which is funded by USDA NIFA. And to see you all here, we have 120 people online at the moment, and we have almost 500 people registered. So there should be a lot of activity today. And so I'm just going to go through some housekeeping before we begin. Well, I wonder if I can make things move. There we go. Okay. There is going to be a recording of this webinar, and it will be available in about a week. We have to edit it, so it takes a little time for us to do that. You can also, on the same webpage, find recordings of past webinars. We did one last year with Jen and Kim, and also we did one a couple of weeks ago, and you should be able to find the recording for that right now if you get inspired after this webinar. For taking questions, we ask that you use the Q&A feature rather than the chat feature. It's hard for us to keep track of everything with using the chat. And so if you put them into the Q&A feature, we can see the questions, and then we can mark which ones have been answered, which ones are still open for us to answer. And if you're not familiar with the Q&A feature, if you scroll over your screen, put your mouse somewhere on your screen, you'll see there's a black bar that appears, and it might be at the bottom or at the top. And there's a little box in the middle that says Q&A. And if you click on there, you can ask a question. And there's an option where you can ask it anonymously too. So if you don't want everybody to know that it's you asking the question, feel free just to check that, and it'll come through anonymously. All right. And our two presenters today are Kim Skirm and Jennifer Lund. And Kim is with the Massachusetts Department of Agricultural Apiary Program. And Jennifer Lund is with the Maine Apiary Program, and they've joined us here today to share their wisdom and knowledge. And of course, everybody's probably at home, including us. I have a cat. I've seen cats in Kim's environment and dogs in Jennifer's, and I have my college-age child, young person living with me. So you'll have to excuse it's not going to be exactly the pristine environment that is common. So. But I'm sure everybody who's watching also has folks in their background. So, all right. So first of all, we have some questions for you. And Kevin, if you can pop up those poll questions for us, that would be wonderful. There we go. Okay, so I'm just going to be quiet for a few minutes. These are not trick questions. There's not a right answer. It's not a test. We simply want to be able to gauge where people are, what people are doing, what you're thinking, so that we can tailor the presentations to your needs and your experience. So please just take a couple of minutes to answer those questions, and then we'll share the results at the end. And I think you have to answer all of them before it'll let you submit them. All right, we have 77% of people who've inputted their answers, so we'll end the poll and I'll share the results with you. Okay, it looks like it's sharing. So as you can see, about a 70-30 split of people who are currently monitoring, but 100% of people are planning to monitor for Varroa mites in the future. So we have an engagement are really looking forward to learning. And most of the people say that they would use alcohol roll. And the question about the threshold limit before you consider the number of, before you consider treatment, and most people answered for that 3 to 9 mites. We will find out during this presentation if that is the correct answer. And most people consider themselves to be somewhat knowledgeable, varying a little bit towards moderately knowledgeable. So that's a great crowd of people, and hopefully Jen and Kim can help you feel more moderately knowledgeable. Okay, so with that, we will move along, and sorry, there we go. So we're going to be talking today about covering a little bit about Varroa mites, and then Varroa mite integrated pest management, monitoring and inspection, and mite sampling methods, and what steps to take after sampling, and then a little rundown of future webinars. All right, so Kim, take it away.
Oh, no, me.
Oh, there you go. Okay, there you go. Jen, take it away.
No problem, no problem. So thank you everyone for joining us today. We're excited to have you all. It seems like it's a very popular thing to be doing webinars now in the current climate that we're in. So I'm going to talk a little bit about Varroa mites, just a refresher from the talk that we gave last week. If you did not check, or 2 weeks ago, if you did not check that out, please go back and watch that. That was an hour and 30 minutes long, full of Varroa mites. And this is just a crash course just to get us kind of a little bit all on the same page. So Varroa mites are small red to brown triangular mites that feed on adult pupal and larval bees. And they're one of the largest ectoparasite to host relationships. That means they're very, very large compared to their host. Next slide. So you can see here, that's about how big the mites would be if we scaled them up to our size. So imagine walking around with one of those on you. We lose our minds for ticks, which are comparatively much smaller. So walking around with 1, 2, 3, go ahead, next slide. Up to 5, 10, 15 of these at any given time. You can imagine that would be pretty detrimental towards your overall health. So go ahead, next slide. So Varroa mites have a 2-phase life cycle. They have a dispersal phase, which used to be known as the phoretic phase, where the female mites pass from adult bee to adult bee. And they're doing this as the bees are touching one another, as they're moving, or as they're coming in contact with one another. So if 2 bees visit a flower, one has mites, one doesn't, those mites can walk across right onto the bee that doesn't have mites. And they can move to uninfested colonies by drift. So bees are very social, just like us. They like visiting other bee houses. And so they will go in and investigate, bringing mites potentially with them. And they're feeding during this stage between the abdominal segments underneath on the underside of the bee on the abdomen. You can see on that top picture, those 2 mites are tucked in right in those abdominal segments, and they're feeding primarily on the fat bodies, a little bit on the hemolymph, but primarily they're feeding on the fat bodies. And fat bodies are really important in all insects because they play important roles in detoxification. So if a bee eats something that's toxic, they'll break it down into non-toxic substances. They also play a role in the immune response of bees to outside pathogens. And then finally, they play a role in backup energy supply. So if there's a food scarcity, they can actually consume part of their fat bodies and live on that for a bit. So that's 3 of the 9 functions that fat bodies have, and those are 3 really important ones. And then in their reproductive phase, the female mites will enter an about to be capped cell and hide in the brood food. She'll start laying eggs once that cell is capped. The first is unfertilized, becomes a male. The next eggs are fertilized and become females. And the average founder's mite would produce 1 to 2 offspring per worker cell, 2 to 3 offspring per drone cell. And that's because the drones take a little bit longer to produce. They have a couple more days of that they're in the pupal stage. And as a result, those mites can produce 1 more offspring during that time, up to 1 more offspring during that time. And it's important to note that 50 to 90% of Varroa in a colony is inside the capped brood cells. So the vast majority are in, if there's brood in their hive, they are in these brood cells. That's where they want to be. That's where they're reproducing. And then on average, mite populations will double every 20 to 30 days. So on average, every month, your mite populations are going to double. So next slide. And Varroa has lots and lots of effects on the bees, including shortening worker lifespan, impairing foraging ability, flight behavior, and orientation. It increases worker drift and robbing. It increases queen supersedure, it reduces sperm production in drones, it reduces colony level honey production, it reduces colony level winter survival, and it increases the likelihood of virus transmission. So this is all research that has been found over the last 20 years of some of these effects that Varroa has just in these hives. So they're really, really detrimental. So next slide. Questions?
Great. Let's see. We have a couple of questions that have come in already. And there's a couple here from Tony Braithwaite, and I'm going to ask actually Kim or Jen if you want to read that, or if it's better for that question to wait until the next until a later part of the presentation.
Yeah, that looks like a waiter one, a wait one.
All right.
And so there's one, Tony, down at the end. When a founder's mite enters a second cell, does she still lay a male first, or can she produce a female right out of the gate, having been mated before in a previous cell? So I think that new research, and correct me if I'm wrong, Kim, that summary paper that just came out did say that they produce a male regardless of whether they've reproduced before. And that's because they want a male in the environment to mate with any other female mites in there. So they're going to produce one, and that's really what's passing on a bunch of their genetic material. So producing males, they can mate with multiple female mites, and pass on that genetic material more efficiently.
Okay.
So yes,
right.
They still do.
All right, and then I think the other questions are probably better ones for us to wait until we get further down into the material, because I think they're going to be answered. So the next topic is Varroa Mite IPM, Integrated Pest Management. And well, whoever is going to jump in on that one, go ahead.
All right, so next slide. So I want to talk a little bit just about integrated pest management as a general decision-making process. So whenever I think about pests and diseases in a hive or anything, even my garden, I use integrated pest management kind of to make decisions. And that's using a combination of techniques to suppress pests. And the first one is planning and managing to prevent organisms from becoming pests in the first place. So making sure, in the case of a hive, your hive is in a good location, that you have really good sun exposure, that it's dry, it's not sitting in the middle of a swamp. All those things set up your hive to begin with, spacing to reduce the likelihood of pest problems. Second is identifying and understanding potential pest problems. So this, taking webinars like this or courses with your local bee schools or bee clubs, going to meetings, learning about what pests and diseases are out there that could potentially impact your hives, as well as learning about what potentially could in the future. So it's not just what's here now, it's could be here in the future. And keeping up to date on that, because if we do get something like tropicolaps mites in the United States, we want to, be prepared and understand those things before they come here and become problems. And then monitoring pest populations, and that means really getting into your hives and looking to see what you have going on in there, as well as monitoring for mites, and that's a really important thing that you can do. Once you get in there, see what's going on, using injury thresholds to make treatment decisions. That means using numbers to make decisions. I've had beekeepers tell me in the past, when I asked them why they treated for mites when they did, this is a true story. I actually had a beekeeper who told me that they had a nightmare the night before about mites, and so the next day they actually went out and did a treatment based on that dream that they had for on Varroa mites. So it's a way to make a decision, but maybe not the best one. And then doing something when you reach those numbers where those mites are getting out of control, reducing your pest population and using strategies that many different strategies, not just doing the same thing over and over, but using a combination of strategies. And then finally, and this is a really critical step, is evaluating the effectiveness of your treatment. So making sure what you did actually did something. It's really easy for us to put in a mite treatment and then walk away and not think about it again kind of thinking that we did something when in fact that treatment failed for any one of various reasons. And so going back and actually making sure that what you did something. And so next slide. So with the goal of IPM, you're not going to eradicate every pest in your hive, but you want to hold them at levels where they're not harming your hive. And then IPM uses all kinds of different techniques, so biological, physical, chemical controls, as well as habitat modification. And so it's not just doing the same thing over and over, it's using a variety of techniques to get at a pest. All right, so next slide. So there is this guide out there made by Cornell University, and it's got this really great little graph thinking about how we can do Varroa Mite IPM. And it starts at the bottom with getting good genetics in your hive, then monitoring cultural practices, and then some of our miticide treatments up at the top. So you want to do those other things before you get to the miticide treatments. Next slide. And so really when it comes to principles of Varroa Mite, integrated pest management is really being knowledgeable. So be knowledgeable, know bee and mite pest biology, so understanding the biology. Be a keeper, so monitor your hives frequently to determine mite levels and compare with established thresholds. Be active, use your integrated management approach consisting of prevention and intervention. And then be prepared. I don't know why that's number one again, but we're going to be prepared and knowledgeable at the same time. And that's establishing an IPM plan for your apiary before the season starts. So thinking about these things beforehand. And then next slide. So is this you, Kim?
Is it? Yeah, again, I was going to say I can talk a little bit. Sorry, I think I threw that little number in on Jen in the last one because my version of doing this is after a softer. Jen is the more hardcore scientific one, so I just wanted to leave it with a fun and warm and fuzzy IPM thought for the day. So there you go. There's your gift for the day, IPM. Okay, so yeah, so this is a survey that we did in Massachusetts of our beekeepers. We've been doing this survey for the last couple of years at the local level. Inside of these bars, you can see the number that you see listed is the number of beekeepers that responded. So these are surveys that are available online. Beekeepers took them, and we asked them basically a question that said, "Did you monitor for Varroa mites?" Similar to what you see in the webinar we're doing now. And you can see we actually did this to see if the grant that we received from the Northeastern IPM Center to do these webinars, in addition to handing out the mite wash kits and stuff we'll show you later, was effective at all. So when we started this in 16, 17, it was just an idea of this could be a tool for beekeepers coming on board, trying to find ways to support them. And still, even if we look at going through the years of this survey, we're still seeing, I mean, right now, right, I would love to see this 100% mark. I'd love to see every one of these bars be all the way to 100%. And even today, I mean, we still had a group of beekeepers who say they don't monitor for mites. So I think we still got a little bit of work to do. And that's part of our job today, is to do a little dance to maybe convince you more that you need to be doing mite monitoring extremely more involved than you have been, or if you've never done it, ways to help you go through this. So it's interesting to see, though, of the beekeepers that aren't monitoring for mites. If we go to the next slide, we'll show you a few more statistics, but some of the reasons why people aren't doing that. And I would love to also maybe get some of that from the questions as well, whenever we get down to it. But Maine, here, I'll let you take this one, Jen. I don't want to speak for the wonderful state of Maine. I don't do it near as justice as Jen does.
Oh, you can do it. We've given each other's talks so many times that I'm sure we can handle it. So for monitoring in Maine, we do an online survey, anonymous online survey as well, and we've been increasing the last couple of years, about from 50 in 2016, 2017 season to 50% of people were monitoring, all the way up to this past season, about 65% are monitoring. So we are seeing an increase, but there's still a third of people who are not monitoring. So that's next slide. And it's interesting because I have 3 years of data on the beekeeper data that they've sent in to me. And so this top part of this slide is the 3 years, 2016, 17, 17, 18, and 18, 19. And N is the number of beekeepers that fall within the category. So our overall losses in range from somewhere between 43% to around 45%, a little bit above 45%. Those that are not monitoring, their losses range from 45% to almost 60%. Those that are monitoring, it's 38, 39% to 45%. Those that are using alcohol washes have their losses overall on average run between 28 and 39%. So by far, those that are monitoring using alcohol washes are having lower losses on average than those than on average than those that are not. And then these other methods of monitoring, things like looking for mites on the backs of bees, or looking for drops on a sticky board, counting drops, or even the sugar roll, the losses for people who are doing those alternative methods are around 50%. And so those methods are definitely not giving beekeepers a good idea of what's going on in their hive.
And if we see down in the, can we go back one more, Jen? If you see down in the bottom here, this is data from the Bee Informed Partnership, which is we're actually referred to them a couple times in this presentation. It's a great resource to go to for seeing data that beekeepers responded to. This is a survey that they also offer online for beekeepers, and 285 folks responded. I call this the New England-ish states because we've included the great state of New York, so you're the ish part of this, but it'll give you kind of an idea overall for people that responded to the question if they monitored for Varroa mites. You can see the folks that obviously did not monitor was much higher, but people that monitor for Varroa mites, unfortunately, the lower is the better. So this would be a much better response, but still, we're not at 100% with any of these. I mean, it doesn't even go to 100%. So we've got some work to do, and it will be our job again today to answer questions and try to help you if you're on the fence about whether you should monitor, or you're confused about what to use to monitor, or the process of it. You should not leave this webinar without those questions answered. So please don't feel shy, and make sure you get that information known, what you need to be supported in this process. So yeah, sorry. Thank you, Yana. All right. So Jen's like, I'm done. As soon as I saw her muted, I was like, oh, that's a universal sign. Look at it. Washed hands. So, okay, here's the Kim show now for a couple minutes. So I can't do Jen justice. But so Varroa mites, the thing about them, I think that makes this a little bit harder for people to understand, is that you've had a chance now to understand about their biology, how it is definitely intertwined with the honeybee biology and the hive management. But the piece that's really missing that I don't see beekeepers oftentimes being able to correlate is how, because they are so intertwined, how we have to manage for these pests just as much as you're managing for the health of your bees in other ways, whether it's requeening the hive or splitting them or putting on honey supers or whatever. This has got to be a vital part of that process. And in order to do that, in order to even start that, you have to understand what the population level is, right? This is our IPM basic. So you need some sort of accurate sampling device to do that. And a lot of people have been really creative and coming up with ways to try to find out how many mites are in their hives. And visual inspections is one of them, and we'll give you a couple of seconds here in a minute on future slides to test your skills on seeing these on the backs of bees. But a lot of people do these pupil tests where they open up pupae that may be in the purple eye phase of development, where their eyes are actually purple, so they're that many days old underneath the capping. They'll open them up, and you can actually, they're looking for mites on the bodies of the developing bees. Now you can see even from this photograph how difficult it is to see this mite that's really, really lightly colored, and it may be a little easier to see this mite, but obviously this one really stands out. So if you were to open this up, it would be really hard to find these two mites, whereas you may only be able to see one, so you would have an inaccurate estimate of the mite population. Also, this is a really difficult thing to do because there's not really good guidelines on how many pupae you should open, what it would look like as far as, if you opened X number of pupae, how that would translate to the overall infestation level of the hive. And more importantly, what if you have no pupae? What if you have no male or drone pupae in the hive to open? So this could be a really not great way for you to monitor for mites. The other way that I see a lot of people view this is that I don't see any mites on my bees, therefore I don't have any Varroa mites. And hopefully we'll go to the next slide. We'll give you kind of a chance to see how this can be really misleading if you're looking for mites on bees. So if you're doing the mites on bee part, obviously if you're sitting still and they're in a photograph, you can see them. If you're doing the pupae, you can actually pull them out. So you could use one of these uncapping forks to pull them out, and you could visibly see the mites on the pupae. There are a range of a lot of different types of sampling devices you can purchase, like this apiary diagnostics kit that the folks in Ohio put together. It's got a lot of stuff in there for Varroa management. The kit from Minnesota Bee Lab that you can purchase, that's for doing a powdered sugar shake. This one I think is one that you can use. Was it CO2? I think in this one, Jen, I think this is from our European colleagues, where they've using like a CO2 to basically knock out the bees and the mites, and that actually kill bees or kill the mites per se. And then we've got a variety of ones that you can use with alcohol or with powdered sugar. For instance, the Varroa Easy Check that you can purchase from Vito Pharma, or this design that I think you can purchase from our colleagues in Canada. I think I've seen that for sale. I definitely saw them at Apimundia for purchase, where there are 2 jars affixed together. And then this is one that our team really likes, a little harder to craft, so we don't give these out. But if you can create a way to, like a sieve inside of a jar, you can obviously make this really easy on your own, purchasing the screen. But there's a lot of things available. And I guess as beekeepers in the beekeeping community, we like to buy stuff. We like to have stuff. We like the gadgets. We like to order from catalogs. And I don't think it's great to have all these things, because if you need to get something that works really well for you, and something that will allow you to maintain some sort of stability in your sampling. So that way you can correlate and compare your numbers throughout doing this in the season. The other thing that I want to recommend, and we'll go through with doing an alcohol wash, we're actually going to take you through the steps of this, is to be consistent not only in the equipment that you use to do the wash or to sample, however you want to sample, but also the materials that you use in that equipment. So for instance, if you're using alcohol, consistently use 70% isopropyl alcohol. If you're using powdered sugar, consistently use powdered sugar versus confectioner's sugar, or versus, other things that you might be using. Not to say that those media are more preferred over others. It's just, if whatever you start using, it's great to stay with that so you can compare the data through time. So if we go to the next slide, let's kind of do a little quick test here of your ability. And maybe if you want to, Yana, we can take a quick break, because I think this will be fun for people to get a chance to view this for a little bit. Because when I do this in person, I have people call out numbers they see, and obviously it might be really hard to do with this group of folks. So we'll give them a couple minutes to stare at this a little bit, and a little cheap primer. How about that? And we can take a couple questions. Is that okay?
Sounds perfect. And maybe what people could do is, if you could put in the chat your best guess of numbers, that would be a fun way of doing it. So great. And actually, I had a question here from Phil Pither. He said, if 50% are living in the cells and doubling every 22 days, isn't sampling mite population with alcohol wash too late and just a prophylactic treatment?
So did you get that, Jen?
Yes.
You're reading it too?
Yeah, I'm reading through. So no, it's we still recommend doing it, because you may not actually need a treatment. So the way the alcohol roll wash, it kind of gives you an idea. It gives you the percentage of bees that have mites on them in your hive, and those numbers will change kind of throughout the season. So if you're starting, for instance, if a beekeeper, which we'll get a little bit into this later on, if a beekeeper did their first alcohol roll in May and they rolled a really low number, that doesn't mean that they need a treatment. So if they're just kind of prophylactically treating, somebody may do something in May when they don't need to. And so that's really what we're getting at, is refining or making those decisions on when you need to treat a little based on numbers versus a feeling on what's going on in your hive. I hope that answered the question.
Okay. And there's another question here from Colette Snyder. Have you found any research on Varroa sensitive hygiene bees, VHS bees, that genetics keep mite count down better?
Oh, I was actually just answering that question. So we're going to talk about that next week, or next, right? Is next one, Kim, no, that one's treatments. And the one after that is the fourth one. We'll be talking a lot more about genetics then. But just as a kind of tease for it is that Varroa sensitive hygienic does do a good job, a better job than other bees in keeping mites lower for longer. Unfortunately, we do find that they're not able to do it all by themselves. So good genetics is great, and that's really important to have in a hive, especially if it's anti-mite genetics of some sorts. But typically we find that those hives collapse just like hives that don't have those fancy genetics. But they do on average keep the mites lower for longer. So.
Great. And actually, this might be a good time to clear up a confusion that someone had from the last webinar. I've always thought that if somebody asked the question, there's other people were thinking it too. So this is from Teresa Laney. She said, I was confused by the last webinar relative to reusing frames from dead colonies. Obviously, no one is going to destroy all those frames. Also, extracting the remaining honey stores is inconvenient, and post-treatment wouldn't be consumable. Since the concentration of viruses is higher in bee bread, would it make sense to eliminate those frames only?
I think, yeah, this is an interesting question. We get a question about destroying equipment all the time, especially when we visit and do samples and then provide, the lab send the results and they see they have a virus panel that's really high or really diverse or both. Yeah, it's tough. This is a situation, especially when it comes to bacteria like European foul brood, American foul brood. It's so much easier, and specifically European, because American, we would have you burn everything. But that's a little easier question to answer, because then if you got only a few cells that are infected, we could get rid of the frame. Usually it would clear that up. There are times we see that it doesn't, but for the most part, it clears it up. When it comes to the viruses, boy, we're still in the dark about a lot of things to manage these, especially as it comes to how to reuse equipment or use equipment. I think we're still really lagging in information around how long they can last without the host, how long they can last on wax, on the woodenware, on your gloves, your jacket. So I think any sort of recycling that you feel comfortable doing as far as trading out equipment is always beneficial, whether you have viruses or not. But I think it would be really hard to say. It's a really good point, though. I think it's a tough question to answer. Maybe Jen's got a better answer, and she can just say yes or no. But
no, we're still trying to figure out how long those viruses live on comb. I would say bee bread, if you do have huge pollen stores or bee bread stores in the hive, that would probably be your best bet to remove that. So if you have a whole frame of bee bread left over and your hive died from really high mite levels, and you are seeing signs of bees with viral symptoms, yeah, you can just pull that and dispose of those frames. But as far as how long it lives on the wax, we don't know. A lot of that research is still being done right now.
yeah.
Great. Well, I think people have enjoyed spotting the Varroa mite. And, we have a whole bunch of responses ranging from 2. Somebody saw 13. Janice Parent saw 13. The average seems to be about 4. So put us out of our misery and tell us how many there are here.
All right. If you scroll to the next slide, I think we'll get the answer there. There you go. Yeah. So you get actually for folks who maybe have just missed it, if you go back, Yana, and go ahead again, we can just show them one last time. So I think the one that people always see first is this one in the center, right? You can usually see that one pretty easy. That's a gimme. This one may be a little harder because she's darker, but you can definitely see that one. Let's see, this one over here, this is getting really tough. So Janice Perrin, this is where you come in. You got a really good hawk eye, so that would probably be one you spotted first. You'd be great at Where's Waldo, Janice. I'd like to have you on my team if we ever played that game. And then this one over here, also a little harder to see because it's at an angle, but you can definitely see it. So a little background about these photos. This actually came from a bee kill that we investigated here in Massachusetts. We call them bee kills because they're suspected to be due to pesticide use. And so we go out, we take samples, and on this particular investigation, the beekeeper was not able to be present. So I was taking photographs of everything that I saw in the hive so I could share that with this individual. Later, when I uploaded these, I noticed some really interesting things when I played around with the photos, and I said, oh my goodness, this would be a great learning tool. So this was one frame, and then on the next frame, the next slide, if we go to the next one, we're really going to give you a test now. Uh-oh, oh no, it told you the answer. Oh no. Well, we just ruined the surprise. Sorry. You're getting 13 mites for Christmas, everybody. There you go. Well, so you can see on here, actually, if we go, can we go back one? Does it let us see it without the mites on it? Oh, there we go. Okay. So if you look on this slide, a little harder to see, but unfortunately we gave you the answer, but if you took a second, you've really got to look for some of these pretty difficult. The whole reason behind this is not only is it fun, obviously, to find mites, but this is not real life, because how many times do you open up a hive and bees don't move? So they're not going to stand still for you and let you try to get a photograph or count how many mites are on them. And the second is, you know, this is pretty unrealistic. You can see just from the response, the variation in the responses of the numbers that people saw. So this is definitely not a very reliable tool to be waiting for to make decisions about mite management. If you're waiting to see mites, you're waiting way too long. If we go to the next, if you go down one, Yana, we might be able to see them. So this is always a favorite here, this double mite. Go back again, we'll scroll through here. Yeah, you can see there's one there and one there. Some of the harder ones, these are some of the easy, well, I guess it's the easier one here. You can see that one really easy. You can see that one really easy. Definitely see that one really easy. This one very easy. So yeah, quite a few mites. And I think, again, this is very hard. Number one, no one is having cameras available this easily in the apiary. Number two, bees are really difficult, I think, oftentimes get photographs of to do something like this. And then number three, not sure this really tells you much, except for that you got a very high mite level in this hive. And by the time you come back to your computer, download these and look at them, it might be way too late for you to actually put on a treatment and maybe do some management that would help. So just wanted to kind of give folks a fun task, let them see that this can be a really difficult thing to do. And if you're using this as a survey tool, it's probably not the best one to use, actually not the best one to use. And there are some much better options available for you. So, but I do always want to cover this because I get this question a lot, and we see it a lot when we visit beekeepers, which is that common response that I don't see mites on my bees, so therefore I don't have a mite problem, or, you know, or my bees don't have mites, right? Everyone else's bees can have them, but my bees are special. They don't have any mites. So it's a very different, it's a very unique way of thinking about it, and I totally get it. Unfortunately, everybody's bees have mites. It's just what level of mites they have and how they're able to manage them, as Jen said earlier. So let's see what we got next. We'll keep going through here. Yeah, so the ways you can monitor for mites, we talked a little bit about, we showed you some ideas on the previous slides. We've obviously, hopefully discouraged you from using the visual monitoring as a big tool. And whenever we've done surveys in the past in Massachusetts, these are kind of the three big mite preferred monitoring methods that people use or like to use, with the alcohol wash or the alcohol roll being one of them, the sugar shake or the sugar roll. There's a lot of folks who use different terms for these. And then of course a bottom board, just having a screen bottom board so mites can fall through along with debris. And I find that a lot of people are sometimes using these in collaboration with each other. So people will use an alcohol wash or an alcohol roll, and they'll use the bottom board to judge. I do see a lot of folks using the bottom board before and after they've done treatments to see if the treatments are working. A good example would be like oxalic acid as a treatment, and we'll talk in more greater depth about treatments in the next webinar. But so some folks are using these not only for monitoring for varroa mites, but also specifically to see if treatments are working after they've been applied. So these two methods, you can actually purchase or create, put together the same physical materials to do them. So you can use the same jar, the same screen, the same lid. Obviously the difference would be this method would use alcohol and this method would use powdered sugar or confectioner's sugar. And you can do the bottom board if you have a screen bottom board, right, in conjunction with these. So these don't have to exist in a vacuum. But I just want to, again, caution, whatever you use, make sure it's consistent. Now, if you've got a big apiary, large apiary with multiple hives, you could be your own researcher. And I actually encourage beekeepers to do this, and mostly for them to see the variation between using an alcohol wash and a sugar shake, and of course a bottom board as well. But in our survey that we did, you could see for the most part, beekeepers, once we started through the years of doing our Northeastern IPM grant, we saw a huge interest in use of the alcohol wash. Well, I mean, huge, we're still a little bit of 50%, but a lot more than had ever used it before, versus before we started the grant and giving them materials for this. So it was really neat to be able to see people start adopting the use of this method. If you go to the next slide, we're going to get a little bit of information into what the data they were seeing as far as the same methods to monitor. And I'll let Jen talk a little bit about this.
Yeah, so this was the data that, again, this is that online survey where I asked beekeepers to give me, fill in all their data on these various different things. And Yana, can you just click forward twice? I think there's 2 hidden things. You have 1 more. And we kind of have similar numbers in that about 1/3 of people are using sticky boards, and some are using multiple methods, so these numbers don't quite add up to 100. So, but 39% of the people, or 31% of the people who responded this past year using sticky boards, and sugar rolls are in between, and alcohols is about 1/3. But you can see those numbers since we started this program also have increased from 11% back in 2016-17 using alcohol rolls. So, good.
So go Maine, woo! All right, so if we look at the data again go back to the Bee Informed Partnerships website, the data is a little harder to find. So you have to go into, I think at the top it says like citizen science or data, and you can click that down and you'll go to, if you're wanting to see this for your particular state, if you're not in one of these states, or if you just want to see it for your single state, like for Connecticut, for instance, you can just choose that. But you will go in and choose hive monitoring and survey data, and you'll see it in there, and you can go through all the different the data they collected. But I chose on their mite monitoring techniques alcohol wash, and what this tells you is how many beekeepers lost bees, percentage of beekeepers that lost hives using an alcohol wash versus not using an alcohol wash. And obviously you see a little bit of a difference here of much fewer beekeepers losing the percentages of their hives versus those that did not use an alcohol wash, and even lower for the alcohol wash, a little higher for the sugar roll, but not by much in comparison. But definitely just kind of want to convince you again why the alcohol wash or the alcohol roll is a little bit better of a method. If you go and choose it for the sugar shake or sugar roll, you'll see a little bit higher levels of loss versus those who didn't use it. Actually, those who didn't use it was much lower. Anyway, so, but yeah, again, good take home here. So you can take a look at that data if you want to see it by state. If you go to the next slide, so I'm not discouraging the use of a sugar shake or a sugar roll. And again, if you have the equipment, be your own scientist. You can totally take the same hive, take 2 samples, do these side by side, compare and see what numbers you get. And you can do it on your own to see if you actually can convince yourself to use this if you're still on the fence. And I think this question is really hard for people who say to me, Kim, I don't want to kill bees. I don't want to kill any bees in my hive. Bees are already struggling enough to survive, and I want something that's really not invasive and it's not going to be destructive to kill bees. And I totally get it. It makes total sense. However, the tough thing about using a sugar shake or a sugar roll in our climate in the Northeast is that powdered sugar, if you do this ineffectively, it can also kill bees. And we do have a thing called humidity up here in the summer, and it can really put an issue when you're working bees to try to get an accurate count. If any moisture gets inside of that jar when you've got your sugar in there, for bakers out there, you know what's going to happen. So it's not going to be as easy for the bees to get it off themselves, much less to get the mites off, which is the whole point of the sugar being in the jar. The sticky board or the bottom board is a great way to check for mites to drop, but it's not a great way to do a full-on long-term assessment of your mite population, especially if you're using treatments or if you're doing IPM in the hive and you're trying to figure out if these methods of treatment are working. This is not the method you want to use to do that for long-term, for pre and post treatment. We talked a little bit about the drone brood uncapping, and then of course visual inspection we went over. So this is my plea to ask folks to really consider using an alcohol wash or an alcohol roll, because, and I'm going to go through the ways that I think we think about this a lot, and definitely the ways that I've convinced not only myself as a researcher, as an apiary inspector, but also as someone that works with beekeepers and manages my own bees, so, and our state apiaries, is that it's cheap. That's always a plus, because beekeepers, we like things that are very economically efficient / cheap. So that checks a big box for people. It's also very easy to obtain, easy to use. It's easy to store. It's durable. And the cleanup of this is fairly quick. And actually the cleanup also sanitizes because they're using alcohol. So it'll kill, should be killing most of the things that could cause issues if you feel like you're going to transfer that. You can use it, reuse it fairly easy. I tend to recommend beekeepers, if you have a couple of hives in your apiary, then you would get a couple of jars set up. So you can just have one jar per hive. And as you go through, pull your sample, it can be sitting there waiting. And then at the end, you can sort of get that gratification of how many mites you had that, of course, are now expired because you put them in alcohol. But at least you can get the numbers and the data in the end. And then if you need to go back and treat, or if you need to do some sort of management tool, you have all the data you need, and you can go through the apiary and, you know, put those treatments on or whatever you'd like to do for management. Then the reliability, we've talked a little bit about this, but it's much better for you to have a tool that you're using, again, consistently, and the media, whatever the substance is you're using to do the sampling process, also needs to be consistent. And we actually see this quite a bit with beekeepers who use the alcohol wash or alcohol roll and interchangeably just grab whatever alcohol they have. Obviously, we don't recommend you to use drinking alcohol and do an alcohol wash, because it probably will not be very effective as far as for you as a beekeeper. But if you do, if that's the only alcohol you have, you know, don't drink it while you're doing an alcohol wash, but you can definitely use it to do an alcohol wash, but keep in mind you would need to purchase that every time. So the best and easiest thing to do is to go to a dollar store near you. You can get all the materials, and we'll go through those next, of what you need to do an alcohol wash in a one-stop shop. It's a couple bucks, and you'll have everything you need in addition to the alcohol. That will probably be maybe not currently available, but maybe very soon available in your local drugstore or dollar store. So yeah, but if we go to the next slide, we'll show you. So we originally had, the goal of these webinars were to show you, to do a video in the field where Jen and I would go through using the state apiaries that we have here in Massachusetts that are managed by the state for educational purposes, to provide outreach education to beekeepers. We were gonna meet up, go through them, have a film crew come and record us actually doing an alcohol wash and showing you sort of where the hiccups that we see most commonly with beekeepers doing this. And given the situation we're currently in, which no one expected, we've kind of had to readjust our schedule. So that's why these webinars changed, and also the process of this. So we'll hopefully be able to do that video, but in the meantime, in case we aren't able, we're going to offer you this. So this is my offering to people that are in Massachusetts. If you do not have an alcohol wash kit, i.e. a jar with a screen and an associated Varroa Mite IPM brochure, we'll be happy to send you one. So you just need to send me your information. I'll mail you one. Probably won't bring it to you given the situation we're in, but the mail, as long as it runs, can deliver it to you. And if you're in Maine, I don't want to speak for Jen, but I think we could probably do the same thing. So if you're in other states, we may not be able to send you an alcohol wash jar. However, if you are in other states and you come to a meeting in Maine or Massachusetts, there's a pretty good chance that we will have these at our booth or a table at the event, and we'll be happy to give you the materials. Jen is like, no way, no way. Look at Jen's face. Yeah. So no, but so if you're in other states that are not in New England or in the Northeast and don't travel to Maine or Massachusetts, we cannot send you an alcohol wash jar. However, I may be able to send you some screen that could go on top of a jar that you may already have, whether it's being used for mayonnaise or pickles or whatever. So this is something that we can definitely think about. So if this is your limiting factor to being able to perform an IPM plan in your apiary, by no means do we want to stand in the way of you being a progressive and fantastic beekeeper. So I want to help you reach your maximum potential, and we will give you hopefully the tools to get there. So this screen is the key. If we go to the next slide, we'll show you sort of what it looks like as far as the brochure. Now, if you want to download this brochure, it is available online through the Northeastern IPM Center, and I'll take you through a couple of slides and show you how to get to it. on the back of the brochure, which will be the first page if you download it, because it says trifold. But if you were to fold it, on the last page, the last part of it would show you the steps of doing an alcohol wash. So this is available to obviously read. You can look at the pictures. Today, you can rewatch the webinar, which we'll go through step by step. And then additionally, if you were to get your hands on an alcohol wash jar, if you're in Maine or Massachusetts or associated states that you could drive to a function in one of our states, those directions are also on the outside of the jar. So we tried to make it very easy for people who are trying to do this or adopt this in the apiary. If we go to the next slide, we'll go through a little bit more of questions. Yes, let's do it.
All right.
All right, I got some questions for you. I got one. Why not assume you have mites?
Oh, I like this question. Who did this question? I like it. Yes, you do have mites. Sorry to deliver that message to you. I feel like it's not a great message, but yes. So it's not about, I think, having mites or not having mites. Hopefully, we've dispelled that myth that everyone has Varroa mites at this point. I'm not aware, and maybe Jen is aware, if there are probably some very isolated populations maybe in the world, and I'm saying it has to be really isolated for them not to have Varroa mites on their bees. And is Australia still doesn't have Varroa mites? Is that the only country?
No, there's a couple island communities too, like so islands off of the Southeast Asia and places like that.
Yeah. Yeah. So otherwise, you all have mites. It's not about assuming whether you have them or not, because you do. But I think it's it's a good question too. It's about what level of mites you have and what that looks like as far as what kind of beekeeper you want to be. And in a sense that, are you going to be a beekeeper to actually manage honeybees? Or are you going to be someone that's breeding Varroa mites and affecting the neighborhood? So, because I think if we don't think about Varroa mite levels and we don't manage for them, we do end up becoming more of an issue for neighbors. So we definitely have them, but we need to keep them below a threshold as part of our IPM process so we can figure out when to act. And I think that's a good question though, because it's something everybody's thinking.
Yeah, and you also don't want to treat too many times. Yeah. So you don't want to treat unnecessarily, because some of these are expensive. Some of the treatments are expensive when you're talking $20 a treatment. And some of them can be hard on your brood and queens, and you just don't want to do that over and over unnecessarily. You also don't want to treat too late or not often enough if you need to, because once the viruses get hold, that's a dead hive. It just doesn't know it yet. And so, so getting that timing down is really, really important.
Yeah, and I think that actually answers Janine Martin's question: why not just treat for mites with oxalic acid and not do the alcohol wash, which kills the bees? So what I'm hearing is that you don't want to not treat and not monitor, and you don't want to just treat without monitoring, but finding that sweet spot of what would be the most beneficial for your hives. So.
Yeah, and we also have a question about the gauge of the screen. So that's number 8 screen, right?
Yeah, I think it's a 1/8 inch hardware cloth. At least that's what our local hardware store uses when I order it. That's what they use in the system, and that's a tough one for people that are looking for that. I've not been able to find it in a really small quantity. I think the thing I've seen that comes the closest to it of it that's a single is from a company that produces screen for sprouting, like sprouts that you would eat, like alfalfa sprouts. I've seen those same screen diameters sold to put on a jar for that, but I've never seen it in a smaller quantity. So this is, again, maybe a limiting factor, and we don't want that to be an issue. So, we'll figure out how to get some screen to you if you need it. Can't send you a big old jar in the mail, but we can definitely see some screen. So.
So let's see. This is a good one. Has there been any new studies on mites and their biology and possibly controlling them or eliminating them? And so usually I point people to the Honeybee Health Coalition's guide for Varroa management, because that is kind of where they consolidate a lot of that information on kind of what's going on in the mite world, the practical stuff for beekeepers to be doing at least. But there's always, always, always new research coming out nonstop about Varroa mites and how we can better control them. There's some research being done on new miticides all the time. Everybody's always testing new miticides, as well as behavioral techniques. And I don't know, what else am I missing? there's always tons of research on Varroa going on.
There's always something. Yeah, the latest thing I think I saw was really interesting. Is I know that some folks got a grant to look at some sort of laser that would zap mites in the hive. Which again, I really applaud all the folks that are thinking about this outside of the box. I'm not sure what that looks like economically, but there are a lot of people really thinking about this in a really intentional way, and that could come up with some novel stuff. So stay tuned. I think the latest thing that was probably really fantastic that I saw that came out of hard research was the multiple mites in the cell stuff. That was, I mean, that's a game changer. I mean, we're obviously past the hemolymph, the fat body feeding, which was a huge game changer, but the multiple mites in the cells, that's super, makes it a little bit harder to think about management. So.
I see Jen has one more question to answer now. From Karen Hayes, is I've heard that the sugar method isn't considered scientific, i.e. it doesn't yield good data. And is there something you'd like to add to that?
Just mostly what Kim was talking about earlier, that it's if you're in a high humid area or an area, if you get any moisture in those jars, it really does impact the results that you get with it. So it tends to be a method that is not as reliable or repeatable for many beekeepers. The other really important thing about the sugar roll is that you have to really shake that jar. Most people are using it because they don't want to injure the bees or kill the bees. But in order to do a sugar shake the correct way, you have to actually be shaking that jar very, very hard, which there was a little bit of research that came out. This wasn't a published thing. I just heard about it secondhand, where somebody looked at the survivorship of the bees after a sugar shake, and they actually don't survive either if you do it the correct way. So if you're really shaking them hard, they're getting knocked around and they do get damaged. So it's, you know, if you're doing it incorrectly, you're not getting the right answers or the right threshold number, or you're not getting the right numbers. You're not getting an accurate read on it.
Yeah.
So.
And yeah, and so one last thing I just want to make a plug for is if you do the alcohol wash, the other awesome thing about it, and on the previous slide about the data, kind of my ways to convince you to use it is that now that you've alcohol washed the bees, you also have them in a preservative. So you could take that same sample and send it to a lab or save it, or do whatever you would like to do with it, or if you would like to come together and everybody share their alcohol washes and everybody count, and kind of like the slide with the photograph, how many bees can you find? It's a way for you to also be able to reuse that and or get additional information out of that sample. So it could be a really nice 2-for-1 if you've already killed the bees. So it lets their body go to other things, which I think is a really great way for you to get more data.
Great. And before we move on, I just want to put in 2 questions here that you'll probably cover in this next section. Somebody said they have trouble getting 300 bees for an alcohol wash from a top bar comb. Usually I can get 1/3 off the comb before they start flying out of the jar. So maybe you can cover that as you're going through. And then somebody else said they lost their hive last week and they want to do an alcohol roll mite test on some of the dead bees, but they can't get hold of any alcohol. So if you have a suggestion for an alternative alcohol people could use as you're going through, that would be great. So.
Yeah, and so I've responded to a couple of those. It is hard to get alcohol right now, so you can use alcohol from your house. I'm a little more liberal up here in Maine with my alcohol. I usually say 1 for you, 1 for the bees. Whatever. However, I don't judge. But the another alternative that you can use is actually a windshield wiper fluid. So you can use windshield wiper fluid. It doesn't foam up when you shake it, and that's important, but it will, a lot of times, especially the winter wash stuff, the blue winter wash stuff has some alcohol in it, and that's usually enough. But you want something that'll kill the bees and the mites, but not foam up. So you can't use dishwashing soap or anything like that. So windshield wiper fluid is a good alternative.
Yeah. And I think that's still fairly available, right? I haven't seen anybody go crazy with that yet as far as...
No, it's not like toilet paper or alcohol.
Yeah. Can you imagine if this included toilet paper as part of this?
And there is still plenty of cheap vodka too on the shelves. I noticed that the other day. So
Jen, this is a theme. How do I not know this about you? I feel like okay. Well, this is I wouldn't know anything about that. I don't think that I was saying I have a glass of soda here, but I don't think that Coke will give you any real results on the alcohol wash. So yeah. All right.
Are you ready for me to move forward?
Yeah, let's do it. Okay, so here is your the best things. These when people ask me, I want to get into beekeeping, what things do I need? A past and hive tool and a smoker and a veiler jacket. These are the next things I always tell folks, and probably the cheapest items you will also buy as a beekeeper as far as tools go. So this is a standard wash tub or a wash bin that you can get from a dollar store. You can usually find them for $1. You may even have one laying around that your significant other will not mind you using gently and bringing back. But also the other piece to this that you need is a measuring cup. So someone mentioned earlier about if I can't get 300 bees, which makes total sense, especially in a top bar hive, could you do a smaller quantity? So we do recommend using a half cup measuring cup, but obviously if you wanted to do half of that, right, you could just use a quarter cup. So you could use a quarter or half cup. I usually tell folks when you go to buy the stuff, and Jen has given these out as kits quite frequently, so she's very aware of probably what's available, but you obviously would get the whole set of measuring cups. So, don't get rid of them all because you may end up using some of those other parts of that. I don't recommend doing a whole cup of sample. That's a lot of bees, but definitely keep the half cup and the quarter cup because those might be two sizes that you could use. So if you did a quarter cup, right, it would be supposedly in a half a cup if you measured out dry weight of bees, dry as individuals in the hive alive, in a half cup measuring cup was around 300 bees. It's not exact, but right around there. So if you did a quarter cup, that'd be about 150, which might get you pretty close to your top bar hive sample if you're able to get that. And then you just see here, there's some regular rubbing alcohol. I think this one is 70% is what you can get, again, what you should historically been able to get at a grocery store or drugstore or dollar store. And then the jar. So this jar is a regular canning jar that you could use to can vegetables. I think that we usually tell people to get a quart size jar. I do have beekeepers that really like to get a quart and a half size jar. So if you can find a little bit larger, because it's a larger volume on the inside of the jar. And so when you put the bees in and the alcohol, it gives them a little bit bigger area to work with as far as seam material. We usually just recommend the quart size because it's a little easier to come by and it's cheaper. You can also, and beekeepers like this, is getting the wide mouth version of this jar. So obviously if you get a ball jar or some sort of canning jar, they have a standard regular mouth, but if you get the wide mouth, it may make it a little easier on you to get the sample inside of the jar, because as we're going to go through we're going to be sampling obviously live bees. So this is going to be maybe a little harder for you to get them in if this is the first time doing this. So if you can find a wide mouth jar, the materials of the jar are up to you. So there are a lot of variations of this design we saw earlier. Some people are putting two jars together. Some people are getting plastic jars. Some people are using pickle jars or whatever they have at home. This is, you can totally be creative, whatever you feel like you can find or have easy access to, or the monetary funds to purchase. The key to this though is making sure you find a jar that has a lid that can be removed. So that can be a solid lid and can be a screened lid, right? Because you want to be able to wash the bees with a solid lid and then sieve the bees through the screen lid. So this is where it gets a little bit more complicated when you're using stuff from home, unless you can design something that can have, both lids. If you have two jars of pickles, right, you might want to sacrifice a jar, but keep one lid, cut out the inside. You could super glue or hot glue the screen in, and so you could use that from home. So there are ways you can adapt this. Don't feel like you need to go out and spend a ton of money. And then obviously people have created, I think I've seen, like Jen, maybe correct me, like a Starbucks cup version of this, where people have done like nested Starbucks cups that are kind of inside of each other. So I've seen people do it with Trader Joe's, like, was it containers for nuts or crackers or something, where they're nested in cookies, or something where they're nested inside of each other. So there's a lot of different variations of this. The big thing though, again, is making sure you have some sort of measuring cup. Half cup is preferred, but if you want to do a quarter cup, a quart size minimum size jar, The rubbing alcohol, that needs to be 70% or whatever you feel comfortable with. If you have 80% or you have 50%, what you don't want to do with the alcohol though is dilute it to the point of no alcohol, right? So you don't want to get a 70% alcohol jar and then dilute it to where it's 2% alcohol, because you want bees to actually, unfortunately, expire. You don't want them to be flailing around inside of the jar. You want to be able to kill the bees per se. So yeah, and then of course some sort of bin or wash tub. The color of the wash tub is also critical. So you'll see this is a white one. Most of them are typically white or light in color. If you do find ones that are darker or black, darker colors, might be really hard to see the mites, so we discourage those. So if you can find a white or lightly colored one, that's what we prefer. So let's go through the process of doing an alcohol wash. So those are the materials. When you go into the apiary, I already have this set up. If you go to the next slide, the first thing to do, right, you're going into the apiary. I usually set up all my materials. So I've got the wash bin, the measuring cup, I've got the jar. I unscrew the lid of the jar. So I just have the jar open and I lay everything near the hive. So the goal is to get it as close to the hive as possible, so you can reduce the stress on yourself, scrambling or dropping something or spilling something, and then also the stress on the bees, so you can be as least impact as far as being in the hive and or getting the best sample. This is a beekeeper that I was working with, and she was kindly enough to model for me here the process. So you're going to open the hive, you're going to go through a couple of the frames. Obviously, we want to get to a brood frame. So this is where this is critical. If we go to the next slide, you can see this beekeeper is going through. She's looking on the frames. The first thing you want to do is get into, as fast as you can, the center of the hive. So, again, we're trying to work where we're least impactful. We're trying to get a good sample, but we also don't want to keep these bees open for hours and hours and hours. So you go and try to get into the center. The goal when you're looking on the frames, if you go to the next slide, is you want to be able to quickly see the queen. So for people who are on this slide, I'll leave this up here for a couple more seconds. Hopefully you can see that queen dead center of this frame. And you want to make sure, I usually try to tell people to make sure the first thing to do is not only to find the frame that you want to sample from, but once you find the queen, then you know that you're not going to use her in your sample, right? And I'm just going to do a disclaimer. You're going to kill a queen. It's going to happen. It's not anything you're going to do on purpose. It's happened to all of us. If a beekeeper tells you they've never killed a queen, they're lying to you. Everybody kills a queen. The thing about it is we want to keep as much as possible not killing a queen. So when you pull out that first couple frames in your hive, you're getting to the center of the hive because we want to find a frame that has brood on it. And we can see on this frame with the cells that are capped and then some of the cells that are open. If we were to zoom in, and maybe some of you can even see, depending on how big your screen is at home, you may be able to see there is not only cap brood with those sealed cells, but also at the bottom and the right-hand side of this frame. But also there are open brood cells where this queen, near the queen actually, you can see eggs in there. And if you really zoom in, you can see larvae in some of these. So we want to find a frame that has open brood, specifically open brood. So the reason we want to do that is if you have not watched, and hopefully you have and remember from the first webinar, open brood and the bees that tend to those brood, nurse bees, tend to be the most attractive to Varroa mites. So we're trying to target those brood frames so we can find out the best estimate of the Varroa mite population in our hive. Now, obviously, we don't want to sample from this frame because the queen is on it and we don't want to kill her. So this would be a frame where you go, okay, great, now we know where she is. If you go to the next slide, we would start putting this frame back inside the hive. So, and that's good because once you've got that situated in the hive, if you pulled out a few more frames while you were getting to this middle section, you may be able to find some open brood on those with something to work with. And that gives you a little bit more security because we know that we're not going to kill the queen. However, the disclaimer here is that you still always look for the queen. Even if you put this frame back inside, even if the frames that you took out are not anywhere near this open hive at this point, and you're going to go through them, you still want to do a really good glance on both sides. And if you have somebody in the apiary with you, have them do it as well. I always have someone check after me because it's just good to see that, to look for the queen. And that way you know you're not going to put her in your alcohol wash. So if we go to the next slide, we've now figured out we've got a frame, we've got some brood on the frame, we know where the queen is, And one of the frames that we pulled out of this hive has a lot of brood on it. But this brood, as you can see in the photo, is mostly capped brood. So we don't want to target that frame. We want a frame that has open brood. So we're looking for a frame that has eggs and it has larvae. Not saying that there aren't mites on these frames, not saying that they would not give you a sample. They would definitely give you some information, but it's not going to give you the information that would be most reliable. And it would definitely not be the best thing to sample from. So we put that frame back inside. We go to the next slide. Now we go, okay, now we can work with this frame. We see there's open brood. We see we've got a nice population of bees. There's also some closed brood. And we do not see a queen on this frame. Now, granted, this is just a snapshot zoom in, but what I wanted you to see is that, well, a couple things. Number one, remember, the bees are not going to wait for you to look for a queen, and they're not going to wait for you to assess the frame. So you got to really get your eyes trained to what it looks like to see where open brood would be, not only in the hive, right, when those center frames that you're going to focus on when you pull them out, but also on the frame itself. And then make sure once you got the queen, you figured out sort of what she would look like, what the entourage around her, the following, the activity looks like when she's on the frame. And then more importantly, the frame that you end up choosing, make sure it's got enough bees on it. So sometimes I find beekeepers will pull a frame out, go through this whole process, and the frame they choose has 20 bees on it. And it's like, oh, yeah, okay, that's not going to work for us. You know, we need at least a good sample size. A minimum, I would say, of a quarter cup, but ideally, we want to hit that half cup mark. So this would be a great frame. So now we've went through a lot of trouble in the hive to make sure we've not selected a frame with a queen. We've got a frame that has open brood. We've got a frame that has a sufficient number of bees. Now, this is the next task. So now we've got our frame. We've got our wash pan or wash bin beside the hive. If we go to the next slide, the process, for some reason, the minutes ago, every time I see this, when the beekeepers are in their hive, just minutes ago, we realize that we're working with live bees. And this moment though, right before we shake these bees in the wash bin, all of a sudden, everybody gets reality check and goes, oh, I don't know if I can do this. These are live bees. There's bees on this frame. And I'm like, yeah, there's been bees on this frame because you've been keeping bees. But this is the scariest moment every alcohol wash, every single one. So you just gotta pull it together in the last moment here if you're doing this. And this is real scary because what's going to happen is you're going to turn this frame on the side. You're going to put it in your wash bin or your wash tub. This happens to be one that I got at a garage sale here locally. But it's, again, just white. It's got a nice opening big enough to hold the side of a frame. This happens to be a deep frame, so it's going to be a little wider. I do want to make a quick caveat. If you're not running deep hives, if you're running mediums or you're running something like earlier with a top bar hive and maybe you don't have a frame you can pull out, there are other ways that you can do this, and we can show you or walk you through it. But instead of at this point you tapping the bottom of this frame into the wash bin and making the bees fall off into the wash bin, you could just take your jar and gently run it down the side of this frame, and the bees would basically fall inside fairly easily. The good thing, if you did that, you wouldn't need the measuring cup, but the good thing about these ball jars is that they have a half cup unit on them. So you could figure out if you had a half cup sample or pretty close to it, or if you did this and you don't have a jar that has that, you could count the number of bees at the end of this because obviously the bees are dead. So you could get your proportion or your ratio that way. So there's a couple ways you could work through this if you're not able to shake a frame. So, but if you're a traditional Langstroth hive, you've got frames that you could pull out. Once you have them, so you want to take that frame that was once horizontal that you were looking at, you want to rotate it to where it's vertical now. And then once you do that, now you're going to gently and as harsh as possible, which sounds contradicting, but you want to gently tap the end of that frame inside of the wash bin to knock off the bees into the wash bin. Now, I find that sometimes people do this really, really, really gently and 2 bees fall off. That's not going to work. But don't worry, they're forgiving. So you get another chance. They will not judge you. So, and when I do this, it usually happens so quick. People are like, oh my goodness, that was really rough. Well, you got to get them off the frame. So you may be, when I say gently, it may be a little harder for some people. What you don't want to do is you don't want to tap this horizontally, because what you don't, if you turn this around vertically where the frame is being held like this versus like it is now, then if you tap this horizontally the way you were looking at it, the larvae or the brood could fall out or be damaged. And we're not trying to damage any larvae. We just want to target these nurse bees that are on this frame. So that's the goal. So once you tap it on the end here, this vertical end, the bees, if we go to the next slide, this is where you got to pull it together, are going to, guess what? Live bees are going to come off that frame inside of the wash bin. And you're still a beekeeper. You're still a good human, but we're taking a sample. So you got to keep reminding yourself when you're doing this, because I see beekeepers constantly going, oh, I'm really nervous now, or I don't know if I can do this part, or this is getting really scary. This is the go time. So you'll notice once we've done the tapping of the frame and bees fall into the wash bin, a lot of bees, I mean, a fairly good number here, are flying back out. And Jen mentioned earlier in her webinar, and we'll talk about it, I'm sure, again, the bees that typically fly out quickly are those foragers. And we do have foragers that are on these frames, right? Because there may be resources on the frames in addition to the brood. And those are not the bees we want to target in an alcohol wash. We want to target those nurse bees. And unfortunately for them, the nurse bees don't fly away like the foragers do. So they're going to become part of the sample. So at this point, you've got them in a great place. You can see it happens really quick. That's the end of the frame in the corner of this photo. So I've tapped them. I've pulled the frame back. Very quickly, you've got to put that frame that you have either back into the hive. If you've gotten really good at this, you'll just quickly put it back in. But when you're first starting out, you might just leave this on the side of the hive propped up. Not on the ground, not laying horizontally flat, but just propped up vertically on the side of a hive. And now, if you go to the next slide, you want to be able to see most of the foragers have flown away. This is seconds after taking the first photo. So you've got a fairly decent, mellowish population of nurse bees ready to be sacrificial lambs for your sample. So if we rotate our bin in the next photo, we start the process of rotating this. You're going to stand this up vertically, and I'm going to take this measuring cup that I've so nicely placed right next to it, right? Because I'm working on the grass right beside the hive. And I'm going to scoop out a half a cup of bees as best as you can, right? This isn't like scooping flour or sugar. You don't top this off with a, with some sort of butter knife. They're live bees, so they're going to crawl out of there. But you can do a pretty good job of trying to feel that space. So if you go to the next slide, you'll notice the process is fairly, and when I'm doing this and taking photos, so I'm laughing at myself because it does not look this easy. When you're in the middle of it, it looks kind of chaotic. But if you just keep moving through the motions of this alcohol wash process, I promise you in the end, you're going to go, oh my gosh, that was not as bad as it felt like in certain parts. So the bees are still alive, right? They're still moving around, but you've done a pretty good job. And I've tilted this for the photo. So you're going to have this up, this is tilted like this at this point. It's going to be firm and horizontal for you to get as much of a sample and get it over to, in the next slide, to the jar as quick as possible. So you can see bees are still going to be trying to come out of this jar or measuring cup to go before they go into the jar, and some may fall out of the measuring cup into the jar while you're doing this. You do notice that I have gloves on here, and I don't keep these on because they help you eliminate stings. You can still get stung through these nitrile gloves. The reason I have them on is for disease management, and we also do a lot of hives. But if this helps to wear gloves when you're doing this, if this makes you feel more comfortable, by all means wear them, because I think this is a critical process. When you get to this step, you don't want to spill this because you've worked really hard to get this half a cup of bees. So spilling it might be a lot more difficult to come back from, for your level of comfort to get back into the hive. So if we go through the next slide, you'll see we're going to pour these straight into the jar, and most of these unwilling bees are going to go in about as good as bees would go in any jar with liquid in it. By the way, missed a big step. We go all the way back. In addition to opening up your jar and leaving the lid off, we also wanted to take a little bit of the alcohol and feel the inside of that jar. So this is critical, because if you're doing this, you notice I'm doing this on the grass or the ground at the base of the hive. If you do this elevated near the same height of the hive, I've seen beekeepers spill these jars and pour alcohol inside of the hive. So just be real careful if you want to do this at your working height, that you keep it where this would not be the possibility of being spilled in the apiary. You don't want to kill any more bees than you have to. So the level of alcohol that you would pour in here, or washer fluid, or whatever liquid that you want to use, is about the same as what's in a half cup measuring jar. So I tend to use the half cup to measure out alcohol, and then wipe it out, and then use it to measure out my bees. If you use these jars, again, the Cannon jars, they do have measurements on the jar, so they'll show you what a half cup looks like, so you can just fill to the line on the jar versus using the measuring cup. So now you're going to pour these bees inside of here, again, as best as this looks like you can. And then if you go to the next slide, this process is fairly quick. You'll notice that you still got bees in the corner inside of the wash bin. They're hanging out. So we don't want to take the whole frame of bees. We're not going to kill them all. We're going to put those back in the hive. So, you know, if you are doing this again, don't pour the bees and have your jar sitting inside of the wash bin, because you don't want to kill any more bees than you have to. So you want to keep this as separate as possible. If you go to the next slide, once you're done with this process, you can see it's still kind of a little chaotic, right? There's bees still crawling out of the alcohol. They'll still be moving around in there for a little bit. In addition to, I've seen beekeepers use a much larger container for the alcohol wash. I've seen beekeepers actually just pour them into the wash bin with the alcohol and have it like 2 wash bins instead of having a jar, which is fine to use, but it makes it a little harder. And realizing certain times of the year when you're doing this, bees may be attracted to stuff that looks like water sources. So I do have bees will often fly inside of these jars and kill themselves inadvertently because they're trying to collect resources. So the less you can keep the opening for, you know, collecting inadvertent bees, the better for you as a sampler. So if we go to the next slide, at this point, the scary part's done. Beekeepers can still be a little worried here, but you can see bees may be flying around. Most of the bees that came from your frame itself are not going to be flying around, because again, these are nurse bees. If you did a good job of choosing the right frame, these bees need to go back in the hive. So now you're going to take this wash bin or wash tub of bees, go to the next slide, and you're going to put those right back into the hive. And so these are the girls that may be sacrificed at a future date, but not today. And this can easily be done by just tapping again the end of that container. So if you read this, steps to do an alcohol wash would take so much less time than the amount of monologue that I have barked at you for 30 minutes on how to do this. If you did the alcohol wash, it would take a fraction of the time than the amount of time that I've spent, again, explaining this to you. And I know that doesn't make a lot of sense if you haven't done one yet, but practice really does make perfect on these. If we go to the next slide, we've got a little bit of tips in here that we'll go over in just a few minutes, but the goal, right, is to start out with the materials as you went in. So you should have a fairly clean wash bin. Sometimes there's feces and things in here, but for the most part, it should be as clean as it was when you started or close to. You've got your alcohol, or you've got your measuring cup that may have a few stranglers hanging around, so you can put those back in the hive or at the entrance. And then you've got a jar filled with alcohol, about a half cup or so. And then you've got obviously a half cup or quarter cup or whatever size cup you want to use of bees inside. So now this is the fun part, if this is fun at all, in this process, is you're going to go to the next slide. You'll take this jar and you'll take what was a solid lid, and a screen lid is underneath that, right? Because the lid's inverted. You're going to put the lid back into the jar. If we go to the next slide, and once you've screwed that on, we now want to give this a pretty good shake. So at this point, I've moved the jar to the top of the hive because hopefully there's not a real concern about me spilling this into the hive. Although this can be really scary if you started this from the beginning. Again, this is still it could be a concern now, so may not always be the best thing to do is to screw this back on the top of the hive. I think I actually screwed it on in this photo and then unscrewed it and said, oh man, I need to get a photo of this step. So if you go to the next slide, you'll see once you screwed this lid on, now we've got, right, a jar of bees and alcohol. That's what we've had from the last couple of slides, but now that it's contained, we want to give this a really good shake. Maybe even a couple of shakes, depends on how hard you shake. The goal is you want to dislodge enough movement where you dislodge the mites that may be attached to those bees. And as we saw in previous photos, sometimes those mites can be in weird places, right? So they could be underneath, in between segments, or underneath parts of the wing or stuff like that. So we want to be able to dislodge them. If we go to the next slide, you can see we do have both of our lids on in this photo. So we've still got the screen lid on, and you can either choose to leave that on or take it off, but the key is making sure you have the solid lid on at this point, because you want to shake that up and down, similar to like you would shake a mixed drink or something like that. And what else would you shake in that manner? I'm trying to think. I'm not a big alcohol drinker, but I imagine martini or so, I don't know, something off the movies I've seen. So shaken, not stirred, right? So once we do that, we'll go to the next slide. You can see I usually leave these alcohol jars with the samples in them to set for a few minutes. And the reason is because it's nice to have a little bit longer, sadly, for the bees to finish expiring, because it does take them sometimes a little bit of time to die in alcohol, in addition to the mites, right, to also die, and then those two to be dislodged from each other. So if you can give this a few minutes to sit. When I take alcohol washes in the state apiaries, I usually take an alcohol wash. I leave it on top of the hive. Meanwhile, by the way, you put the hive back together. So you put the frames back in, you've gotten any other straggler bees that are in piles or on the ground or on equipment back in. You close the hive, you've got the hive cover on, and you're gonna leave your samples sitting there. Also, I use this as a way, maybe to not, it's not a great thing, but like an instant gratification before I leave the apiary. So I usually set these up on tops of all the hives that I sample. And before I leave, it's the last task to go through every one of these, get the number, record the data, and then have some time to think about, do I have what I need to manage bees if I need to do some sort of management based on this value. So it gives me a little bit, and I can keep moving through the apiary in the end to make those hard decisions. So if we go to the next slide, once you've done that, you're at the end of the apiary, you want to go through these samples. You basically would take the solid lid off the jar. This would leave you with the screen lid, and you want to sieve that through. So this would be the point where you want to use that wash bin again. You want to pour out all the liquid that you can. In addition to leaving the bees inside of the jar, you also want to give it a pretty hard shake vertically. I shake it pretty good from the vertical motion to get as much liquid out as possible. And people always laugh because people get at this step and they go, I don't want to shake this very hard. And I'm like, well, the bees are already dead, so you can shake it as hard as you want. They do not mind. So, it's easier for you to shake it. I would shake it as hard as possible, because you'd be surprised at how many mites get dislodged if you don't shake this versus if you shake this pretty well. If you go to the next slide, once you've shaken that, you should be able to see mites. Oh, there's my dislodging step. So all the liquid's poured out in the wash bin. Going to give it a couple hard shakes. We go to the next slide. You'll see now, if I pour those bees back inside, so I've already done the alcohol wash step, but I use this to show people sort of what the mites look like versus what the bees will look like. So the mites in your sample to identify are these black little dots. And if we go to the next slide, I'll show you kind of an up close view of what they look like. So the black little specks in the corners, you can see them. If we were to count the number of mites in this sample, this is again from a dead out hive, but not from the one I was sampling in another apiary in other photos, but you can see there's actually 52 of them, which is a huge number of mites. Your threshold for Varroa mites in thinking about treatment should be between 3 and 9 mites, not 52. So this is much, much higher than a treatment threshold. Actually, at this level, and I'm not sure if Jen would agree, but this is probably past the point of no return. Even if you did a mite treatment, I'm not sure if this hive can come back at this point. It's pretty high. Another issue that we had in a similar hive in this apiary, if we go to the next slide, I'll show you, it can get even more. So you thought that was a lot of mites. If you see this alcohol wash again, zoomed in, we'll go to the next slide. There's 141 mites. So this is definitely a goner, which is tough. But I show you this because I want you to see what a lot of mites look like. Unfortunately, it's much harder to show what a few mites look like because, right, these little specks are teeny as far as in the sample. And if you only had 1 or 2, you'd have to zoom in really close. But I guess I also want people to see that this may not be what you see in the first time you do an alcohol wash. I think people get, if you've been using the bottom board to sample for mites, this may be what you see every day. I mean, you see a lot more mites on a bottom board drop potentially than you would in an alcohol wash. So these two things may not correlate in what you may previously have been using to do a sampling for mites. The other thing is that, again, I want to stress this, the bees they're expired, they're dead. So if you want to take this sample and you want to send it off to a lab, or if you're not sure if there's 141 mites in here, then you can send this sample off, for instance, to the USDA Beltsville Bee Lab. in Beltsville, Maryland, and they will, maybe not during an epidemic of COVID-19, but they will, under usual circumstances, analyze the samples for you and give you some information about what level of mites you had in your sample. So if you have watched this webinar and you go, there's no way I can do this, I'm super nervous, I don't think I can go through these steps, you can also take a sample of bees and send them to the Beltsville Bee Lab before you even go through the steps of the webinar, if that's something that seems a little bit more doable. If we go to the next slide, I want to leave you with a couple of good tips. So the first is, remember, your threshold is 3 to 9. So if you do an alcohol wash and you get 1 mite, then by what research has shown us and the Honeybee Health Coalition resources that have provided us is that this is below the treatment threshold. So what I usually tell beekeepers is if it's less than 3, then at this point I would continue to monitor the hive, which would mean additional alcohol wash samples. And we often recommend doing them at least once per month. I do have some beekeepers that have done them as often as every day. I don't know how they have bees after that, doing it that often. And I obviously have beekeepers that do it once a week, but usually once a month is a really good level to do an alcohol wash. And it's nice. I think people can help with a calendar. You know, I do this on the 1st of every month, or, you know, the 31st or whatever. It seems to be a little easier for them to have consistency. The other thing I want to leave you with as far as a couple of tips. So if we go to the next slide, I'll show you a couple of ideas, the things to keep in mind. So remember, if you're doing an alcohol wash, you want to get a frame that has open brood. We're targeting nurse bees. So we want those bees that are caretaking for the brood. So some people may be thinking, ooh, what if I don't have any nurse bees? What if I wanted to do an alcohol wash on a package of bees, right? That just have adult bees, workers, and a queen. You can do an alcohol wash on a package of bees. I probably would not recommend it until you get some brood, because that would be a good way for you to also see what level of infestation your whole hive is at. So maybe wait the first month after getting a package and installing them. Also be mindful that a lot of places that sell package bees may treat the bees, and they're required to treat them by law when the packages are shook from hives. So they may have already had a mite treatment. So there may be a little bit of preventative measure. We'll talk a lot more about treatments in the next webinar, and we'll give you some tips for package bees. Otherwise, you always want to look for those brood frames to sample and to get bees from. But remember, look for the queen, and then look for the queen, and look for the queen, and have somebody else look for the queen. Call Jen, have her look for the queen. I'm waking her up. She'll always look for the queen for you. She's like, I don't think so. All right. The next thing is, if you do this, we do find that people that rinse the samples a couple of times can get different results. So it does a little bit of a learning curve to figure out kind of where the mites will hang out. I often will look inside of the jar and open up the jar and look underneath the lid. I will also look on the walls of the jar, because sometimes if you're really taking the sample and shaking it out, they may not always come out of the jar with the liquid, so they could get stuck inside of the jar. So it's a good idea to investigate the inside of that. If you don't feel comfortable doing that, or don't feel like you're at that point where you know what to look for, you can just do another rinse and repeat. So you can pour all the liquid back into the jar. You've got the screen, you can do the shake part again, and then you can dump them out again into the wash bin. So you can go through that process. This is a very common question that we get a lot, which is, how many colonies in an apiary should we sample? How many times should I do this? Most of the beekeepers we work with that are hobbyists have 1, 2, less than 5 hives. You know, we recommend that if it's less than 10 colonies that you do them all. If it's more than 10 colonies, if you're a commercial beekeeper or someone that's running 50, 100 colonies, you do between 3 and 5% of colonies. And that's also from, and correct me if I'm wrong, Jen, from the Honeybee Health Coalition guide as well. So we're getting that information from that group, which is by far the go-to gospel and Bible for Varroa mite management. I just want to leave you with these 2 pieces. So this is my quick soapbox for people that have endured listening to me for this long. Number 1, bless your heart. Thank you for coming in. I know this is a lot to listen to. Number 2, practice really will improve your accuracy. And I know that sounds ridiculous because we're telling you to kill bees to get a number. But we're telling you to do this because if you don't figure out what your treatment level is, or what your threshold is before you do a treatment, and more importantly, and I see this so often, if you don't figure out what it is after treatment, what's the point of using a treatment? you need to know if you're going to use an alcohol wash or any other mite monitoring device, you need to know what the level is, because you may not need to put a treatment on, right? Good IPM, and you need to know if the treatment that you used or treatments in your plan worked, or else you may need to do additional management steps. And this is where I think the real big piece was missed in doing Varroa mite washes and or sugar shakes or any sort of mite management, is that after treatment sampling. That really is critical to this. So the same steps you would use to do alcohol wash before you decide what treatment to use are the same steps that you would use after you've decided treatments or a process, whatever management you would like to use, to do another sampling to look for the mites. If you use an alcohol wash, you do a treatment, and then you use a sugar shake, right? Be careful, because that's not the same sampling. So I've seen a lot of beekeepers do that, or vice versa. They started out with a sugar shake. They said, I don't want to kill any bees. I want to do a sugar shake or sugar roll. They do some sort of treatment because maybe that number tells them, you know, it's 20. And they do a treatment. And then after the treatment is done, they say, oh, wow, I just watched this webinar. Now I'm going to do an alcohol wash. Be careful, because I think a lot of people can get really confused and caught up if you see that. So yeah, just be really careful in doing those things. I think, what else do we have on here? I think we definitely have some time. We may not have much time for questions, but yeah, thank you for listening to all that rant. I feel like it's much easier to see that in person. If we get a chance, we'll do the video.
Yeah, no, that's great, because people can play this back. So we will have a recording of it. You can come back in a few days and watch that over again carefully. So what I am going to suggest is we were scheduled to finish at 2:30, and we're at 2:37, and I want to be respectful of people's times. We actually have some great questions. So what I'm going to suggest is that we go through the last of the slides, and if Jen and Kim are happy to stay on for a little bit longer, we can answer some questions live, and then we'll also, Jen and Kim will go through some of the questions that can be answered in typing, and those will be posted. Those will be posted later. That might not be done till the end of the webinar series. We haven't decided how we're going to do that yet, but that is my suggestion. So yeah, so I don't know if Kim or Jen want to go through the sampling exercise in the next few slides. We've got some upcoming webinars and some last details for you. So Kim, do you want to go through this?
Yeah, so a quick thing. So I put this in because we do this in our workshop whenever we do the mite workshops during the winter and aren't able to sample bees. If you're sitting at home and you're thinking, I don't know if I can get out to my bee yard, maybe it's snowing outside like it is here, you can actually go get, these are lemon head candies in a plastic container. You could use beads, you could use pom-poms, whatever you have. You could do your own sampling exercise where you measure out a half cup of them, put in some red things. You could use candy nerds. You could use anything that would be much smaller, about the size of a Varroa mite. Put it in and do your own sampling to sort of go through these steps at home. That, you know, a space that feels maybe a little safer, a little easier to navigate than the bee yard doing this for the first time. And since we are all sitting at home, this might be a fun way for some groups to do some games to see what numbers they can get to share this. So if we go to the next slide, we also want to share with you, we do have beekeepers that really pride themselves. This is one of my beekeepers here in Massachusetts that has kept every alcohol wash that she did that season and was really prideful. You know, her thinking was, if I'm able to keep these, that makes me feel like at least I'm making a difference. And also, you know, she was really happy about all the mites that she's killed in that container. So yeah, whatever gets you through. So a couple of announcements. We got questions there. So once you've done a Varroa mite alcohol sampling, not only is it important to record the data, so we recommend you having a notebook, either that you're keeping for apiary management notes in general, or if you don't have anything like that, you can put it on your phone as a note. You can also leave a voice note on your phone, or if you don't have a phone or device in the apiary, you can actually take a Sharpie and write it on the lid of the hive. So if you've got a hive that's got any sort of aluminum lid or metal lid, then you can just take some alcohol later and wash that off and just keep them for the whole season, date it, put the number that you got. In addition to your own personal recording of the data, we highly recommend you to visit the Mite Check website, who's also one of our partners on the grant. They maintain a website that has real-time data for beekeepers who are importing their mite count data all over the country. And you can zoom in by county. For instance, this is Norfolk County in Massachusetts that we zoomed in, and see that, you know, someone was reporting the average mite level was 5. So that also gives you some information about what your neighbors are seeing as far as their mite wash numbers as well, which is really important, right? Because we're all keeping bees together. And as Jen has went through a lot of the biology that they can transfer very easily from bee to bee and within the landscape. So you need to know what your neighbors are doing and what their levels are as well. So if we go to the next slide, we have been giving out some Varroa mite sampling kits in Massachusetts. So if you're a beekeeper in Massachusetts and you would like one of these kits, we'll be happy to send you one. This is basically allowing you to take the sample that you just got from the apiary in the alcohol wash, and using the same sample to put it inside of the bags and send it to the Beltsville Bee Lab, so you can have them either check it for additional pathogens, like for instance for nosema, which is a gut pathogen that infects bees, or you obviously could send it to other labs, but these are available if you're interested, and if you live in Massachusetts, we can send you one, or yeah, probably send you one at this point. If we go through, again, I want to make a plug for the mite workshop that we do in collaboration with the University of Massachusetts Amherst. Hannah Whitehead, the extension agent. We've been doing these day-longs where you come in person. We'll give you a whole bunch of goodies. You can see there on the slide. We'll also, beekeepers are teaching beekeepers. So this is a really powerful way for you to not only share the knowledge that you have in managing mites and managing your apiary for Varroa, but also to learn from other beekeepers that are locally doing the same thing. So it's really a great opportunity if you're interested. We also want to make a plug for our last bit of webinars. I think if we can go through, we've got 2 more. You can join Jen and I. We will probably not be looking that dapper, but I don't know, Jen, you look pretty, it's the same picture, right? Yeah. Oh, I think you're muted.
I think I might be in the same outfit, so.
Oh, awesome. Well, I definitely am not in the same outfit. Sorry. But yeah, you're welcome to join us for a couple more of these. We did change, again, the format around a little bit. So the next one's going to be on April 6th. It will be for 2 hours. We'll talk about management tools. So we're going to go through all the different tools / treatments / chemical, non-chemical, preventative, and intervention type things that Jen has been talking about in IPM. And also we've been sort of building you up to learn more about. And then finally, we'll go through the most important kind of culmination of this webinar experience for you will be taking home your very own IPM plan. So come to that webinar for sure to bring your ideas and be open to doing a little homework during the webinar. Okay. Jen's EAS plug. I'll let you make that, Jen.
So this one is a little bit in limbo right now because we're not quite sure how this is going to play out over the next couple months, but we are going to be hosting in Maine this summer, hopefully the first week of August, the Eastern Apicultural Society meeting, their annual meeting up in Maine. So it'll be a good time. We have lots of speakers. We'll be covering mites a lot, and there'll be a lot of in-hive demonstration. We're actually going to put hives right outside the building that the conference is hosting, or that we're hosting it in. And so it's a good time. Check it out. Registration will go up once a decision is made whether we're going to actually hold it this year or not. So keep posted.
And we have some final questions for you, similar to the initial poll that we did. And I'm just going to be quiet for a couple of minutes to ask you to answer those questions. And then we'll go back to answering some of the questions that came in during the webinar. So I'll just be quiet for a minute or two. Okay, so we have, okay. So you can see that people are feeling moderately knowledgeable and very knowledgeable after the webinars, which is exactly what we want to hear. 100%, well, actually 99% of people are planning to monitor for Varroa mites in the future. And it seems like we have some conversions to alcohol washes. Is the most accurate method, which is the following survey tools is the most accurate with alcohol roll. And the question for the threshold limit, 3 to 9, is the correct answer, and that's what most people chose. And then this question about which products you use. I think this is something that Jen and Kim are going to use in the next webinar. So that was for our information kind of question. And do you have a Varroa Mite IPM plan? And it looks like it's 50-50. So hopefully after the next couple of webinars, that will be closer to 100. So thank you for taking the time to do that. It makes a difference to us in making these webinars useful to you. So I need to make that go away. There we go. Just to remind you that if you want to see a recording, it's going to be posted in about a week. I'll also send an email to everybody who's registered, and you'll get a link to the recording. And if you are a researcher or you're interested in working with researchers, we have a website on our website called Find a Colleague, where you can give information about who you are and who you're interested in collaborating with and what skills that you bring to the table. And please feel free to post a posting on there. And we also want to acknowledge USDA NIFR, who provides funding for the Northeast IPM Center and also some funding for Jen and Kim to work together and for these webinars. We are deeply grateful for their funding. And thank you for joining us, Jennifer and Kim. And we're going to switch back to, we can probably do a few questions before we need to close up. So Jen, I know you've flagged some of the questions as they've been coming in, so I'll let you take it away.
All right. So we have how much alcohol do you put in a jar per half a cup bees?
I usually try to do at least half a cup of alcohol. Like I said, you can either use the measuring cup to measure out the alcohol before you get started, or you can just, if you've got a canning jar, you can see there's like a half cup delineation on the side of the jar. I mean, obviously you can use more than that too. If you got like a 1.5 quart size jar or one of those really Costco size pickle jars or something like that, you know, you could use way more. But yeah, just you can also reuse the alcohol. So we didn't talk a little bit about that, but yeah, you can get really thrifty, especially now in today's world. You may need to reuse that alcohol.
Yep. So there was also just a comment, can we address the fact that nurse bees are less likely to fly? So shaking into the dishpan and waiting might be preferred. And I think we covered that if you let it sit for, 5 or 10 seconds, those bees that fly out are likely your foragers, leaving behind the nurse bees who are reluctant to fly. So this is a good one. How should we check the effectiveness of treatments applied late October or January, particularly oxalic acid vaporization, when it's too cold to open the hive for a mite check?
This is a good one, huh? Sorry, I'm trying to see the text. I was trying to scroll back up.
So I usually just recommend, if you're doing a treatment that late, do not open up to do a mite wash, and just hope that it was effective, because there's really no way. One thing, I do have some beekeepers who will monitor the mite drops on the pull-out bottom board at that point, just to get an idea of what they are seeing for drops. But it's hard with those late treatments. You just can't get in there to take a look. So
yeah, sorry,
so
Jen, I was trying to read it. Sorry.
Oh.
Yeah, no, I think that's perfect. Yeah, I mean, there becomes a time, right, when you're on a fine line between, like Jen is saying, opening the hive and damaging the brood and causing more damage versus, doing a treatment. I mean, I think especially in winter, in the climates that have like a full winter condition where you freeze, or right now where we got snow on the ground, you may not be able to really assess whether that treatment was effective. And that we're really gearing that comment towards the season when the bees are active and you can assess it. Because if you use a treatment, it is always a great idea. And a critical part of this is to look to see what the sample was after the treatment. And that's what we find people miss the most as far as doing a great IPM plan. But yeah, if you aren't able to, if you don't want to cause more damage opening the bees up, I think that's a great suggestion. Just waiting.
Yep. And so does the Beltsville lab test for viruses?
Unfortunately, they do not test for viruses. So there are some labs that are available for virus testing. I think we've got one on our website at MDAR listed that we use for our virus testing. That's a state vendor. Do you have any? I don't know if there's any on your website, Jen. There may also be some on the Apiary Inspectors of America website. Oh, I can check into that. But yeah, they will not do viruses. They will test for nosema and varroa. I'm not sure if they do tracheomites as a common evaluation.
Not anymore.
Yeah, so not anymore. There was a question about tracheomites too, and I think that's a good one to think about, is that this may, as you become more expert in monitoring for pests in the hive, it will help you later on if you keep managing bees to translate what you learn and how you behave and creating an IPM plan to other pests that come up. So while we're targeting this specifically for Varroa, these are skills that you can use for any future pest that we have managing honeybees.
Yeah, and the question about tracheomite was actually really interesting. So we actually do a lot of sampling for tracheomites, not in the last 3 or 4 years, simply because we don't find it any longer. So the USDA Beltsville lab would traditionally test for tracheomites, varroa, and nosema from alcohol samples. So they would take off the heads of bees, pull out the trachea, put them on microscope slides and look. And they found that over the last 10 years, they're just not, maybe even longer, about 15 years, 10, 15 years, they haven't been seeing tracheomite. They may find one hive here and there, one off here and there. And the reason we actually don't see it anymore is that several of the mite treatments, the Varroa mite treatments, actually treat for tracheomite as well, which had led to an interesting situation a couple of years ago. We got a message from one of our apiary inspectors from another state, and they had huge losses due to tracheomite. So they sent off samples, found lots of tracheomites, and all the apiary inspectors were like, "Oh, send me samples. We never see it, so we'd love to have some for our kind of display, so that we could show beekeepers what they look like, because we just don't see them routinely anymore because so many people are using the miticides that kill tracheomites and varroa mites." And they had this huge loss because many of their beekeepers switched over to using oxalic acid vaporization as their only treatment method, which does not kill the tracheomites in the trachea. So it released tracheomites, they got a foothold, and then actually caused a huge collapse of their beekeeping industry as a result. So kind of one of the reasons why we talk a lot about IPM is rotating those things that you're using in order to prevent making other problems, I guess, is the short story on that one. So the question was why we don't talk about tracheomites as much, and it's because we just don't see them. They're not really in the system anymore because we're treating for this other mite, which also covers that. So we had another question about how long after treatment do you retest?
Yeah, so we're going to talk a lot about treatments next webinar, and we'll go through the labels of these treatments so you can get an idea of the treatment time period. But typically, like for instance, with something like oxalic acid, where the treatment is instant and there's no additional, it's not like something that you're putting into the hive that has to set to go through as a fumigant, a long-term fumigant or something like that. You can do a test fairly quickly after treatment. You do want to give the bees time or the mites time to drop, so you want to at least give them time. I usually tell beekeepers to wait at least a day after oxalic acid. It gives you a chance to recover if that was traumatic, and also a chance for the bees to recover. But Jen, maybe you've got some different stuff for that. If there are treatments that you're putting on, I always have beekeepers, whenever you go to take the treatments off to remove them, if they're physically putting in a treatment of some sort, when you go to remove them, that's a great time to do an alcohol wash because you're already in the hive. So you might as well do a one-stop shop while you got it open.
Great. So we should make this the last question. I don't know if there's something that jumps out at Jen, but is there one that you're bursting to answer? Maybe I'll just bring this up because Will Callum has written, I'm going to caution an alcohol wash as the gospel. The beekeeper must be cognizant of the flush of hatching brood. Test one day early of a major flush and you may miss that you in fact do need to treat. Unfortunately, research through a major flush of hatching brood can disguise the balance point between treating and not treating. Do you want to comment on that?
Jen, I'm watching Jen's facial expression. I'm not sure. I feel like that's if we're this is the social cues you miss when you've been in your house for so long.
Yeah. So there is always some kind of, there will be differences in your hive. So if you are in a population increase in the hive and you have tons of brood, so early in the season, your thresholds may actually be different. Your treatment thresholds might be different. And a good place that actually lays this out is the Honey Bee Health Coalition's Guide to Tools for Varroa Management. In that they talk about, so if you have a huge brood population, most of your mites, you're not going to be seeing those in your mite rolls. And so your threshold might actually be, you might want to consider a lower threshold somewhere around 1%, 2% as opposed to 3%. And so, I encourage everybody to take a look at that guide. It's the Honey Bee Health Coalition. If you just Google it, Honey Bee Health Coalition, Varroa, it'll come right up. And that's something that they do talk about that during there, is that you can actually, depending on the time of year and what's going on in your hive, your numbers, you may want to consider different thresholds. And so, yes, I agree with the comment that there may also be some, taking into account what's going on inside the hive is very important.
Okay, lovely. Thank you. Well, thank you everyone for joining us. I hope this was a productive use of 2 hours. And actually, in the time that we've been on the webinar, we've had a beautiful snowfall here. So it's gone into a winter wonderland. So I'm hoping might be the same for the rest of you. And we'll be having another webinar 2 weeks from now at the same time. And you should be getting an email from me with a link to the recording for this webinar. And we look forward to seeing you in a couple of weeks. And please stay well and have everybody in your family be well and healthy too. So, okay, bye.